Hello, everyone. My name is Osman Mahboob, and I'm a senior at Florida State University, majoring in interdisciplinary medical sciences, specializing in public health policy and administration. I'm in my second year of being at the Roper Laboratory at the FSU Department of Chemistry and Biochemistry, and I will be presenting my poster on the time-dependent measurement of extracellular ATP release from islets of Langerhans cells using microfluidic chip technology. This poster is the first stage of a larger project that hopes to eventually gather valuable ATP release data applying the use of microfluidic chips. The interest in ATP release from islets of Langerhans cells stems from the innate physiology of beta cells and their simultaneous release of insulin and ATP into the islet microenvironment. The released ATP has been postulated to serve as a positive feedback mechanism, boosting the overall insulin release response. And although a broad correlation has been observed between the release of insulin and ATP, the dynamic nature of ATP release, including any oscillations, is unknown. And so to establish an effective system of measuring nanomolar, or in other words, really small concentrations of ATP from islets, I had to optimize a commercially available ATP assay kit, which allows for the measurement of a broad range of ATP concentrations. In the results section, there are 3 graphs which have been experimentally determined to depict the relationship between ATP concentration and the relative strength of the emitted light. The large graph in the middle displays the result of reacting 100 microliters of 25-fold diluted ATP assay with 100 microliters of 5 stepwise diluted ATP concentrations. This was performed in triplicate and showed minimal standard deviation. And so the concern here is that for microfluidic applications, this is a very large volume, and therefore I had to scale down the volumes and adjust the dilution of the enzyme assay and determine whether or not I could derive similar standard curves with linear regression lines on a log-log scale. Fortunately, as observed by the 2 smaller graphs, I was able to obtain reproducible results. The figures below are related to the microfluidic chip that was fabricated specifically for future experimentation with murine and human islets. The chip is designed to allow for the assay to flow over the islet cells and allow for us to collect any potentially released ATP secretions from the outlet of the channel in the chip. The images on the top right here are depictions of the standard curve determination process and the chemical reaction between luciferin and luciferase, which is the bioluminescence reaction harnessed by the ATP assay kit used in this project. A cool fun fact is that this same reaction is used by fireflies to produce bioluminescence in nature. And so based on the established foundations from this study, we hope to move on to manipulating environments in which we can measure nanomolar extracellular ATP secretions and determine time-dependent trends in its release. To close, I would like to thank Dr. Roper and PhD student Mr. Wesley Eaton for their support and guidance throughout this project. Thank you.